Hi, hello. Welcome to the Physiology podcast. My name is Nicholas Verhoeven. I'm a PhD student in molecular medicine, and I hold my master's in exercise physiology. I've been a scientist for a number of years, and I also have nutrition science training during my undergrad. Now, with that introduction out of the way, today's podcast is a really interesting one that's actually based around a mystery. And I've kind of loved these physiological mysteries because, as a physiologist, as what I like to claim myself as, uh, you have to know basically everything that there is about the body. That's kind of the goal of a physiologist, to understand the workings of every single tissue, the different hormones, metabolism, kind of be a jack of all trades and have a base understanding so that you can really kind of interweave your understanding of one system with another system. And in this podcast, this particular topic is based on a hormone that very few, as a matter of fact, I haven't seen any people discuss this hormone, which I suppose isn't all too shocking because it's a complex hormone. It's something that it's not as simple as insulin, as I'll explain throughout this podcast, but it's something that is incredibly important because we focus so much of our attention on insulin and fat loss and its impact on fat loss. And I do have content that's already come out now discussing how insulin relates to fat loss. And it certainly is a fat storage hormone, but it is incredibly singular and insular to look at insulin and then say, well, if you have high insulin levels, then you have greater fat storage and less fat loss. While that, of course, then jumps to this conclusion that then the ketogenic diet, which is low in insulin because carbohydrates stimulate insulin release from the pancreas, that the ketogenic diet is then a superior diet. And as we've seen, that's not the case. I mean, I've shown this through a multitude of different studies, and I'll have all those videos linked for you so that you can check them out for yourself. But it's pretty consistent that the ketogenic diet is a great diet for fat loss. It's a great diet for weight loss, but it isn't superior in any way in those two measures. And this, of course, flies in the face of this idea that insulin is the reason why we can't lose body fat. So then that got me thinking, well, what might be some of the other reasons? Why would keto not have this advantage? I mean, beyond kind of the physics of calories in, calories out and whatnot. Like what's a hormone that may be uniquely stimulated by the ketogenic diet that's not potentially stimulated by other diets that contain carbohydrates? And this is something that's incredibly important because keto gurus, in general, people that espouse the ketogenic diet, if you're talking about Dr. Berg or if you're talking about someone else, Dr. Berg is just the first person that comes to mind. It makes sense that these people would focus on insulin, and I don't hold anything against them in that regard, but I think it's it's incredibly short-sighted to just look at insulin and then just assume that the ketogenic diet has this benefit because it has lower insulin, when again the measures just don't pan out that way according to multiple studies. So I did a bit of digging and I ended up finding this hormone that is called acylation stimulating protein. And so what I'd like to do in this podcast, and if you'll bear with me, you'll allow me to nerd out a little bit and kind of tell you a little bit of the story of how I discovered this hormone and kind of the science behind it, because this was pretty nerve-wracking. It made me stressed just reading the studies. It's an aspect that in physiology you have to be able to read literature and read into it to a point where you may not have an absolute answer just by reading one study. You may need to read 5 or 10 different studies to get more of a comprehensive literature review on a particular subject, especially if it's not very well known. So that's what I did for this particular hormone because there were a lot of conflicting studies. So if you'll allow me, I'm going to kind of nerd out and explain my process for doing that. And I'm going to explain essentially two things. Why keto gurus don't discuss ASP, so acylation stimulating protein. What is ASP? I'm going to leave a few details out mainly because that's things that are going to be covered next week. If you're listening to the podcast, I would ask that you go over to the video on YouTube and check those out, because those are going to be kind of discussing the mechanisms of ASP and whatnot. But I'd still like to discuss a lot of what's unique about this ASP hormone and why I think that a lot of people don't discuss it. And it's, I think, for pretty good reason, to be honest with you, because it is incredibly confusing. If you don't want to stick around and you just want to jump to different sections, I'll have timestamps so you can jump to the section that you're most interested in. Okay, so keto gurus don't discuss this hormone. Why is that? Obviously, all of this is just speculation, but I imagine that a lot of people I'll be honest, I don't feel like a lot of people on YouTube are qualified to be talking about this kind of stuff. And I'm not saying I'm the only one that's qualified. And as I'm going through my education, I'm learning more and more and more. But I think that is one of the benefits of a formal education in things like molecular biology, cell biology, physiology, nutrition, that you can integrate all of those things and be able to, as Physionic is supposed to be, this macro to the micro, from the big picture down to the very microscopic, the subcellular level, be able to explain these things to that level of detail. I think that's imperative to be a fully educated human being when it comes to learning the body. But I digress. The point is, I think that a lot of people just aren't qualified. If they have a chiropractor background, I'm not going to name any names, but if they have a chiropractor background, I just don't feel that they possess the knowledge to be able to interpret studies in the intricacies that they need to. Like, if you don't know, for example, what a BCA assay is, or if you don't know what an immunoprecipitation experiment is, like, how can you read those studies that use those experiments to try and come to a conclusion? Just going off of like WebMD is not sufficient to get a full education, and that's what Physionic really is about, trying to bridge that gap so people can have a better education. But again, I digress. I don't want to go down what Physionic is supposed to be and what it will be in the future. But the point is, I think that some people aren't qualified, and they just avoid the topic. They either avoid the topic because, and I'll explain this, why it's so difficult to discuss this particular topic. You'll find out why. The other thing is, I just don't think that they encounter it, because I don't think that there's a whole lot of research on this protein/hormone. It can be really complex. Again, I'll discuss the complexity in just a little bit, but if you look at endocrinology, which is just one subset of what a physiologist needs to understand, insulin is a hormone, right? So endocrinology is the study of hormones, essentially, and their different actions. So insulin is one hormone, and it has what's called an endocrine action, as in it gets secreted from a particular tissue, pancreas, and then it gets released into the bloodstream, and then it moves into the bloodstream and goes to different tissues. That's all fine. That's great. That's pretty easy to track. You can just look at the bloodstream and see, okay, we see a rise in insulin. However, there are other hormones. There are other hormones, and those hormones actually have other actions, and insulin may as well. And these actions are called, instead of an endocrine action where you secretion and then it goes to another tissue and acts on that tissue, like the pancreas secreting insulin and then it affecting, let's say, muscle cells. Another way that hormones can act is through a paracrine action, where a tissue will release a hormone and then it will affect kind of nearby cells of that tissue. And then there's also an autocrine action, where a tissue will, or a cell within the tissue will release a hormone, a protein, and it will then feed back right back onto that very same cell. So let's say the a fat cell secretes a hormone, and then it will then just turn around and bind the very same cell and then cause a reaction. And then there's also juxtacrine ways of going about endocrinology. Juxtacrine is when cells actually touch each other, and the way that they touch each other with these receptors or with these different molecules, it forces the two cells to then kind of interact, and they change, and that is an actual action. That is a part of this whole hormonal understanding. And then finally, there's also some research on intracrine action, which is where a cell doesn't necessarily release the hormone, but it produces the hormone, and then it acts already within that same cell without being released, that it can have action. So the point is that endocrinology is an unbelievably complex topic. It's probably the one that gives me the most headaches when I'm reading research, because I have to go down so many different avenues to make sure that a hormone isn't doing something through some indirect mechanism, like maybe it's stimulating a different hormone and that's having an action. So it really is a mystery. Like, you are kind of like an investigator trying to figure out, okay, well, if I read this paper, that's great. This research group came to this conclusion. Fantastic. But there's something off about their conclusion. Okay, now I need to figure out what it is that is off about that conclusion. Is it some other hormone that's having an effect? Is it maybe that their conclusion is completely wrong? Do I, if I'm looking at the data, do I come to that same conclusion? So there's a lot of different things that you have to consider, way more than what I'm explaining here. So here's the thing, though, with ASP and its comparison to insulin. So insulin has this really simple, elegant endocrine action where it's, you consume carbohydrates, and when you consume carbohydrates, then you have this release of insulin into the bloodstream. Insulin flows through the bloodstream, and then it affects all these different tissues. Let's say fat tissue, it can be muscle tissue, whatever it might be. And that's all fine and good. It's really easy to track, right? You consume carbohydrates, you just take some blood measurements, and you see over time, okay, insulin is starting to rise. So we know that it's having some sort of an effect on insulin secretion. Great. Or at least insulin presence. So that's fine. But the problem is that with ASP, that's not the case. We don't, across several studies that I read when I was looking into this, is that ASP is much, much more complex. In certain instances, you do see a blood level increase in ASP. Actually, the very first study that actually discovered ASP, they did some of these experiments, and they fed people either carbohydrates or they fed them fat. And they found that with carbohydrates, there was no rise in the levels of ASP in the bloodstream. However, with fat, they did see an increase in ASP levels. And I'll go into what exactly ASP does once I get to that section, but I'm just trying to explain the complexity here. So that was fine. Okay, you got kind of a clear relationship there between the different nutrients. However, what actually ended up happening is I read a few other studies, because I looked at their data and I thought, this doesn't seem extremely convincing for a number of reasons, kind of statistical reasons that I'm not going to go into for this podcast. But then I decided, okay, well, I'm going to read into a few other studies. And what I was really surprised to find is that not all of these studies actually agreed. There was a very well done study where they looked at ASP levels, and they fed people carbohydrates let's say 100 grams of carbohydrates, 100 grams of fat, or 100 grams of protein, just in isolation. And I think it was something like that, within 100 grams or like 70 grams or something along those lines. But it was very consistent across the board, which is fantastic, because that then allows for real accuracy when you're actually getting the data. And what they found was really interesting, that they didn't see an increase in this ASP in the bloodstream. So that's obviously conflicting to the other study, to the very first study that discovered ASP. So, of course, that got the wheels turning. Okay, what's going on here, right? And that's where this mystery starts to really come into its own, because you start thinking like, all right, well, what's going on here? Like, how can we explain that one study has this response and then the other study has this other response, and they're measuring essentially the same thing? And after reading multitude of other studies, kind of all over the place, some of them were in one camp and others were in the other camp. What I ended up finding out is that it's very much about the methodology of how they're actually measuring. And this is why I took a few stabs at people that don't really understand the research, or they're not necessarily qualified to read the research, is because this is the kind of stuff I'm talking about. Like, you have to really critically think about, like, if you have a multitude of studies and like one grouping is saying, oh yes, ASP rises in the bloodstream, and the other grouping is saying that ASP does not rise in the bloodstream, you have to think, like, what's going on. And what I eventually came to, one of the denominator, the actual factor that I found, was that the methodology was different between the studies. And as one group got more and more specific about their methodology, the other group was sticking to this one methodology and getting the same result. So it turns out that in certain situations, and why it's so much more complex than insulin, is that with insulin, you can just measure the bloodstream, cool, you see a rise in insulin, fine. You can do that with ASP to a degree, but actually ASP has a lot of, why I introduced this earlier, an autocrine and paracrine action, where it's actually secreted and it doesn't necessarily find its way into the bloodstream. What it actually does is it feedbacks onto the same cells that secreted it, autocrine, or it can also be released and affect nearby cells of usually the same nature. So for that, obviously the methodology has to be a lot more exact. You have to actually put probes next to the cells that are going to be secreting it instead of measuring the bloodstream, because there is a distance between the actual cells, and then there's like an extracellular layer along with an extracellular fluid, and then that feeds into the bloodstream. So if you end up using probes into that extracellular fluid, suddenly you end up finding out, oh, snap, there is an increase in ASP. But if you were to measure the bloodstream, you wouldn't see that increase in ASP. So there you go. There's this local reaction that's happening, and that's why you need somebody who has some training in endocrinology to be able to really tease that difference out, instead of just saying, oh, well, look, there's just this study that contradicts the other study, so therefore it's just not the case. It's you have to figure these things out. They're essentially little mysteries. So why I'm pointing this out is, one, because obviously the complexity, but that then implies that there is a difference between ASP and insulin, that you can't measure them the same way. Insulin, you can measure the bloodstream, like I mentioned, but ASP, you would have to specifically add probes inside the tissue and see if there are any differences after, let's say, the fat consumption and carbohydrate consumption. And the thing is that studies have done that. And I'm actually going to be covering one of those studies that have looked at a lot more specifics on how ASP works and how it functions, the different mechanisms and whatnot. So ASP feeds back onto the cells that secrete it. Okay, so now allow me to kind of go into what is ASP, which is something that, if you've clicked on this, maybe this is what you're really interested in. So ASP is a hormone that seems to be, at least based off of the research that I've read so far, and I'd certainly be going more into this, and I'll update as I go more into the literature. ASP is secreted from the fat cells. So unlike insulin, which insulin is secreted from the pancreas, from the beta cells of the pancreas, the ASP is released from fat cells. But then what is that mechanism? Why does ASP get secreted? And it gets secreted, so insulin gets secreted when carbohydrates enter the bloodstream. You consume carbohydrates, let's say you consume some sugar, or even if it's like a whole grain, doesn't matter. It gets absorbed into the bloodstream. The bloodstream has a rise in glucose, which is the carbohydrate, like the blood carbohydrate, and that enters the pancreas, and then the pancreas releases this insulin, and then insulin goes into the bloodstream and it rises in the bloodstream, and it starts to bind all of these different cells, all these different tissues, again, an endocrine action. And when it does that, all these cells then respond by opening themselves up and allowing specifically the glucose to go into the cells, which then lowers the glucose that you consume, the carbohydrate that you consume, the blood carbohydrate, back into the cells, and therefore lowering the blood concentration, obviously. So that's how insulin works kind of generally. But the thing is, it's not really stimulated by fat. I mean, it sort of is. Like you see a small bump in an increase in insulin with fat consumption, but the primary mechanism is through carbohydrates. And that's where this idea of like the ketogenic diet, people have been saying, oh yeah, okay, so the ketogenic diet has this effect. It's superior for fat loss or weight loss, either one, because of this lower insulin, because it's not going to be secreting, because pancreas isn't going to be secreting insulin because you're cutting out carbohydrates. Great. Okay, we found our solution. But again, like I mentioned at the beginning, that doesn't actually bear out in the literature, as I've shown in many studies. So then what's going on? Is there something unique? And that's, going back to that original question. And it turns out that yes, there is this hormone called ASP, and there may be others that I still need to investigate. But ASP at least has a similar function. It functions a little bit similarly at the beginning to insulin, but it's actually stimulated by fat. So when we consume fat, our cells package those up into chylomicrons. Those are little packaging vessels with the fat inside. Think of them like UPS. And then they dump these chylomicrons into the bloodstream. Eventually, they usually go through the lymph system first, but eventually they find their way to the fat cells. And at the fat cells, this is where the fat cells start to take up all this fat. So when they start taking up the actual fat molecule, the nutrient that you just consumed, that actually triggers the fat cells to then start releasing this hormone, acylation stimulating protein. So ASP is then secreted. And with this secretion, then you have this effect where you're getting, it's sustaining the levels in the bloodstream, and which is the endocrine action, because ASP, which I have not investigated yet, but ASP does have effects on other tissues, which is something that I'd like to get into at some point. But it binds, let's say, the pancreas, so it can have effects on the pancreas. And believe it or not, actually, it also stimulates insulin, or it helps stimulate insulin, but not anywhere to the degree that carbohydrates do, from my understanding. So it does have an endocrine action, like I mentioned, but what's of most interest to us right now, and what people that eat keto, is that it actually has an autocrine or paracrine action. And I usually focus on the paracrine action. So the fat cells taking up these fat molecules, these fat nutrients from your diet, and then it's secreting ASP, and some of the ASP is going to the bloodstream, and some of the ASP is then just turning around and just binding other fat cells nearby, which then stimulates these fat cells to take up fat. I'm not going to go into the mechanism of how in this podcast, but they take up fat, and then that ends up leading to a decrease in lipolysis as well. So not only do they increase fat storage, but they also feed back and force the cells to downregulate their breakdown of fat, which, oddly enough, or maybe not oddly enough, is exactly what insulin does. Insulin does the exact same thing. The mechanism for how these hormones do that is different. And I will have content coming out on that. But overall, the point is that ASP, while it may not rise in the bloodstream, it does rise in the local tissue where the fat molecules are ending up. And when it does so, fat storage increases and fat breakdown decreases. Now, fat storage is about equivalent between insulin and ASP. They are about as powerful as one another, which just makes me smile a little bit, because people focus so much on insulin, and yet there's this other hormone that is much smaller, and that's why I call it the little brother of insulin. But it's so much smaller, and yet it also has the exact same effect and to the same effect size, at least based off of the studies I've looked at so far. So fat storage increases to a similar degree as insulin. However, ASP is a little bit less potent in terms of stopping fat breakdown. So overall, however, the effect is about the same. How it employs that effect is different. So, well, I can tell you real quick how insulin does this. Insulin affects a variety of different enzymes, but two enzymes specifically, where insulin will, it's been secreted into the bloodstream and then it binds, let's say, fat cells in this situation, and it increases an enzyme called lipoprotein lipase, LPL. You'll hear about this a lot over the coming week as I release content on this particular topic. But LPL is an enzyme that cleaves the fat molecules that are inside the chylomicron that you've consumed, those nutrients, and cleaves them to an acceptable form for the fat cells to take up. So obviously the greater activity of this lipoprotein lipase in this context, and that's really important because somebody who is educated on lipoprotein lipase might come back and say, well, okay, that may not be the case in muscle tissue. Okay, so I just want to point this out, and I'm not going to go into that either, but we're talking specifically in the fat cells in this context. So the lipoprotein lipase, if you have an increase in that activity, that means that more of those fat molecules can be cut and processed to be ready to be taken up by the fat cells. So insulin increases the activity and the amount of this LPL. However, it also, on the back end, it impedes fat breakdown, and fat breakdown is run through one enzyme, although there are a number of enzymes, but one of the primary enzymes people focus on is this hormone sensitive lipase. So another lipase, another enzyme with the name lipase, but it's called hormone sensitive, and in this situation it's sensitive to insulin. So when insulin binds the fat cells, it downregulates this hormone sensitive lipase, so you have less of these fat molecules inside the fat cells that are being released into the bloodstream and to then be metabolized, presumably. So that's how insulin functions, and you'll have to stay tuned for how ASP functions. It's slightly different, but it's really cool. So I guess I'll wrap this up. I've been talking for a little while now, and there's certainly more details that need to come out on this particular hormone, but in conclusion, the data is hard to come by when it comes to ASP compared to insulin. You have so much data on insulin, and again, that's why I don't necessarily blame people that just focus on insulin, because you kind of have to be in the field to really understand that there's a lot more going on than just insulin, although insulin is a major player. So because it's a local hormone effect of ASP, it's really hard to study. So studies are going to be slower coming out on it. So unlike insulin, which has an endocrine action, that being released, and ASP technically does as well, but in the context of fat storage, it's more, we focus so much more on the paracrine and autocrine action. Which, as insulin increases in the bloodstream, it affects multiple different tissues peripherally. So ASP, like I mentioned, does as well, but there may be, it may not be associated with increases in blood levels of ASP. And finally, ASP increases fat storage within the fat cells, just like insulin, but the mechanisms are different, which are, like I said, aren't outlined in this podcast because I'd like to save that for another time. Okay, well, hopefully you learned something from this, and I hope to have the pleasure of speaking with you in the next one. Have a good one, guys. See ya.